I'm going to talk generally about differences between genders or between genders in humans, between sexes in animals, and describe our animal model of autism spectrum disorder or ASD. In the last 20 years, the incidence of a variety of neurodevelopmental disorders, but especially autism spectrum disorder, raised tremendously, and it is now 1 to 2% among the children, newborn, full-term children, because it is higher among premature infants, etc., and I'm not going to go into that. But what is really surprising is not only the increase in the rate, which is because of better diagnosis and a better ascertainment. What is surprising is we don't have really, like many neurodevelopmental disorders, we don't have at hand something to really change the situation. And all the educational programs improve the behavior, but the children with ASD continue to be youngsters with ASD and continue to be adults with ASD. And the services actually grow with the children. Services for adults were non-existent 10 to 15 years ago, and now they are slowly developing in different countries. Now, what is ASD? It is a problem of communication and social development. It has repetitive motor movements and seeking for specific sensory stimulations, excessive interest in very small items, organization, and difficulties in adopting changes. There are a variety of neurodevelopmental problems that accompany ASD, like attention deficit hyperactivity disorder, mainly in males, epilepsy in both genders, and anxiety and depression, mainly in female children with ASD. So we have two different types of behavioral or neuropsychiatric disorders, ADHD, which is an outside type of phenomena, and depression and anxiety, which is an inside type of phenomena. And yet, one is common in males, and the other is common in females. And there are many different symptoms between male and female infants with ASD. Here you see Here you see the rise in the prevalence of ASD with time. Now it is around 2%. And in addition to that, if we take the fact that the preponderance of males versus females is 4-1, imagine how high the incidence of ASD is among male boys. And we really don't talk about that, but 1 in 20 or 30 boys has ASD symptoms. So it is not surprising that this is a real problem which is caused by genetic, environmental, and epigenetic factors interacting in a complex way we do not understand yet. But there are prenatal and postnatal or early postnatal environmental factors which will also raise the incidence of ASD. For example, maternal diabetes. The offspring of children, the offspring of maternal diabetes are 2 to 3 times higher in terms of the incidence of ASD, both males and females. And there are several drugs that would produce it, especially valproic acid, which is an anti-epileptic drug, which we used in our animal experiments, and I will come to that in a couple of minutes. Now, because of the clinical differences, we know now that the methods we use for diagnosis of children with ASD of both genders are inappropriate. And we have to adopt, if we use the EDOS, which is the most commonly used method to diagnose ASD, we have to adopt it to females versus males. And this is something which is not generally recognized in many other neuropsychiatric diseases. We use the same method of diagnosis. And here, in the last couple of years, actually, we know that the cutting point for females using the same diagnostic tool has to be much lower than for males. So here you have an example of a severe neurodevelopmental problem which has very distinct differences between the genders. And the differences are not only in clinical presentations, but there are many other differences. And since we are combining, or we have to look on genetics and epigenetics together, I will try to show sex differences not only in clinical presentation, which we know, but also differences in genetic susceptibility. Females need a higher, what they may call, abnormal genetic load, polygenic, to show the symptoms than males. And not only that, but in siblings or relatives of girls with ASD, the ASD in the siblings is more severe than in siblings of boys with ASD. And remember, this is a disease with a strong genetic cause. Twin studies, siblings of autistic females also have more severe ASD than males. And males with microdeletion of SHANK1 have high functioning ASD, while females are normal. Is therefore ASD a sex-linked disorder? Actually not. But if we look at chromosomal aberrations or chromosomal abnormalities, there is actually a higher prevalence of ASD in children who have a lower proportion of a higher proportion of X. In XO, Turner syndrome, we have 3% of children with ASD. Again, sorry, it is a lower proportion. Only one X chromosome, 3%. In Klinefelter syndrome, XXY, 10% of the children have ASD. And in XYY, there are 20%. So when you have more Y and less X, you have a higher chance to develop ASD. So that Y chromosome is apparently a risk factor for ASD. And there are hormonal statuses as well, which I'm not going to go into. In terms of the brain itself, which areas are or were shown to be affected in children with ASD, we have basically several places that were shown to be affected. The prefrontal cortex, where we have more synapses, increased dendritic arborization, and abnormal concentration of neurotransmitters, especially higher dopamine. In the cerebellum, we have decreased number of Purkinje cells, and in the hippocampus, we have increased cell apoptosis. The hippocampus, which is the area in the brain where you have throughout life the formation of new neurons. So we, therefore, in our animal studies, in our studies in mice, chose to study the prefrontal cortex because actually the prefrontal cortex is relatively large in the mice. There are several animal models of ASD, genetic models in mice and in rats, and environmentally induced. And valproic acid, which we chose as the drug that will induce in our model ASD or autistic-like behavior, because we don't call the differences in behavior of animals really the name of the disease. It is a behavior that mimics the human disease. We chose ASD, we chose valproic acid, because valproic acid is one of the known drugs that increases the incidence of ASD in children as well. And the literature cites 3 to 5% of children born to women that took valproic acid during pregnancy to be affected by ASD. So it is a 3 to 5 times higher prevalence than in the general population. Valproic acid is an anti-epileptic drug that on one hand is well tolerated, and it seems to affect similarly in terms of the pharmacologic effects, male and female. And, however, it is a very well-known teratogen, and it is the highest teratogenic drug among all the anti-epileptic drugs. So if we want to induce ASD, we should do that at times where valproic acid will not induce congenital malformations. Because if it induces congenital malformations, and it could be in 20, 30% or more, then ASD is not real ASD. So we chose 2 models. One of them is treatment on day 4 postnatally. And we have to remember that the first 10 days of mice and rats, the development of the brain is similar to the last trimester of the human brain, the fetal development. So this is a period where you might be able to induce behavioral changes or neurobehavioral changes similar to those that can be induced in utero in the 3rd trimester. So this is the first model. And the second model is the prenatal treatment. But again, prenatal treatment after the time where valproic acid can cause congenital malformations, including exencephaly or neural tube defects, meaning from day 12 onwards. Day 12 of pregnancy, and if we are talking of in mice, we are talking of a length of pregnancy which is between 19 and 20 days. So it is actually during the 2nd trimester of their pregnancy. The mechanism of teratogenesis or teratogenicity of valproic acid, and this is why apparently valproic acid induces neurodevelopmental changes and ASD, One of the most important mechanisms is the effects of valproic acid on histone, being an histone deacetylase inhibitor. So here we have a drug which is actually an epigenetic modifier. And this is actually why valproic acid is so popular to induce ASD in animal experiments. We thought that if we have a drug or an agent that will induce demethylation of DNA, like valproic acid, so what if we use as a counterpart and a possibility to avoid the changes done by valproic acid, a substance that will cause methylation of DNA. And there is nothing better than S-adenosyl methionine, which is our natural substance that donates methyl moieties to the DNA. So our studies were an interplay between valproic acid and SAM, and of course the outcome was measured by developmental studies, by chemical or biochemical studies, and by molecular studies. So I would also add that valproic acid is used as a drug and SAM-E is used as a food additive. And it is sold over the counter, and many people take it as a food additive to help, I don't know exactly what, but perhaps to help to keep a better balance between methylated and demethylated DNA. But if this is true or not, nobody knows. And as I said, our working hypothesis was that VPA will induce ASD-like behavior in the rodents, in the mice, and SAM might prevent or at least diminish the symptoms of that ASD-like effects, of these ASD-like effects. Now, to diminish symptoms, one has to give, or at least thinking about the need to administer these two substances very close to each other. So this is why in our postnatal studies, we treated 4-day-old mice offspring with valproic acid by intraperitoneal injection, and on days 5, 6, and 7, we added by intragastric lavage SAM in the amount of 30 milligrams per kilogram per body weight, but this is really not interesting. And we performed behavioral tests at adulthood. Of course, we measured weight and growth, etc., which I will not go into. And in addition, on day 60, we sacrificed the animals and performed our biochemical and our molecular studies. Most studies before they're done on valproic acid as an agent that produces ASD were done on males. And of course, because there are clinical differences between male and female children, we decided that we are going to look on both genders. So we studied separately male and separately female mice. So the behavioral studies. You see here the 4 methods we used for our behavioral studies. Number 1 is the open field study, which measures locomotor activity, repetitive behavior, exploring behavioral and anxiety-related behavior, which are all symptoms of ASD. Number 2 is elevated plus maze. You see it here. And this measures mainly anxiety. And remember, anxiety is more common in girls with ASD than in boys. Number 3 is social interaction test, the 3-chamber test, which we see here in number 3. It is the interaction between the mouse that was treated or not and a stranger mouse. And number 4, which is a measure of learning and cognitive rigidity, is the water tree maze. And let us see the data we obtained from the different studies. Number 1 is elevated plus maze that measures anxiety. So if we look here at females, more head dipping is less anxiety. Less head dipping in the open limbs of the maze, less head dipping means more anxiety. And if you see here, you see that in the females, why is this not working? Oh, here we are. In the females, valproic acid induced severe anxiety. And this was also corrected by SAM in the right. VPA plus SAM, normal, actually similar to saline or to SAM alone, which you see on the left side. In males, we have the same thing. So our model induced anxiety or increased anxiety in both genders and was corrected, this behavior was completely corrected by co-administration of SAM. Memory analysis, which is in children more affected among girls than boys, we see that valproic acid only reduced the memory in females and not in males. And in females, it was corrected. You see here valproic acid, the green line, and the saline, the VPA plus SAM is the red one, which is actually a correction of the anxiety, of the memory problem. So here we have something which is in human, typical of girls, in mice, typical of female mice, and corrected by SAM. Open field, which is a sign of sociability. Again, valproic acid induced in males higher grooming frequency, and which means that actually this is a sign of disturbed sociability, and SAM added to the valproic acid corrected sociability in both genders. Again, a sign which in females, they are more social than male infants with ASD. Here we got the correction for both genders, for both sexes, and again, SAM corrected all the behavioral analysis. And here we see the last issue is the mice in the 3-chamber. I mean, we're introducing a new mouse, and when we look at males, valproic acid reduced sociability, and in females. And it was corrected by Sam, of course. And in females, valproic acid did not reduce sociability, quite similar to what we find in girls. And again, there is no reduction in sociability. And we studied the oxidative stress in the brain because it is known that there is an enhanced oxidative stress in the brain of children that died and had ASD, or adults, and in many experimental animal studies. And we found increased oxidative stress, more common in females and higher. Actually, the oxidative stress is higher in females than in males. And again, this was corrected by co-administration of SAM-E. As to the genetic studies, we use the Encounter neuropathology panel, which measures 770 genes in the brain. Remember, at day 60, and treatment was on day 4, 5, 6. And we found that valproic acid induces a variety of changes in gene expression. And you see here down in these two circles, the changes in gene expression among males and among females. In females, more genes were statistically significantly changed. But we thought, since we don't know what it means, that you have a gene that was upregulated or downregulated with 20%, but it's still, in comparison to controls, but it is still significantly different. So we decided to use only the differences of gene expression of 50% or more. And this is actually from clinical judgment that differences in heterozygotes are clinically negligible and you need much more reduction or elevation of the gene. So we were left with 15 genes in females and only 3 in males. And again, the expression of these genes, as all the others, was corrected. Most of these genes, their expression was corrected by SAM-E. SAM-E did not produce changes or significant changes in gene expression, while valproic acid did produce. SAM-E, however, corrected the changes in gene expression in both genders. And in females, it corrected them in 75% in males and 52% correction in females. Here is the heat map. And here are some examples of the genes that were drastically changed by VPA, and their expression was corrected by the addition of SAM. These are neuroinflammatory genes, neurogenesis genes, and general biological genes, genes that are known to be associated with ASD in human as well. So, if we summarize, then exposure to VPA on postnatal day 4 caused autistic-like behavior in male and female mice with some behavioral differences between genders. Increased oxidative stress in the prefrontal cerebral cortex, this is where we produced all our studies, was manifested by increased malondialdehyde. This is a sign of lipid peroxidation, and increased antioxidant and the enzyme activity, but again, this was also corrected by SAMe, and correction by SAMe was in many of the genes whose expression were changed by VPA. So in this model, we could demonstrate that the concomitant treatment with SAMe to VPA-induced ASD mice would alleviate or prevent ASD in these animals in both genders, and we could demonstrate many of the differences in the clinical presentation in both genders in children. Now, we continued to what we call prenatal studies, because I have to say that most of the widely used mouse models with valproic acid-induced ASD were by prenatal administration. And prenatal administration was always during days 12 to 13 or later, and not earlier, again because of the limitation of congenital malformations that can be produced by valproic acid. So what we did, in marked difference from the previous studies, where we treated in the first week and studied the effect on days 50 to 60, much later, so we could actually explain the effects as epigenetic changes. And let me tell you that we studied similarly the liver of these animals on day 60, and we found no changes. And the changes were found only in the brain. Here, we were interested to see what happens if we treat the animals on day 12.5. Again, the same things. Valproic acid, SAMe alone, or valproic acid and SAMe. We say 24 hours difference. I mean, valproic acid on day 12, SAMe on day 13, 14, 15, given to the mothers and exposing the developing fetuses prenatally. And here we studied the brains on day 1, post-delivery, and found, to our surprise, that SAMe this time produced tremendous changes in many genes in the brain. Again, the frontal half of the cerebral hemisphere. And here you see males, all the red dots are changes in genes, upregulation or downregulation, statistically significant. You see it in the males, you see it in females. Again, more genes were changed in females than in males. And again, valproic acid alone actually prenatally did not change gene expression. So here we have the opposite. Valproic acid postnatally would induce significant changes in gene expression corrected by SAM. Here we have SAM that produces significant changes in many gene expression, in many genes, in the expression of many genes, and valproic acid, nothing. And the combination of valproic acid and SAMe corrected the changes produced by SAMe. So here, again, we have a different story, but the antagonistic effects of valproic acid, which demethylates DNA and SAMe that methylates DNA are shown here as well. And we don't have yet the complete behavioral tests on day 50 and 60 of this offspring to see if they exhibit autism or not. But we have a follow-up in terms of weight and neurobehavioral development up to the age of 1 month. And surprisingly, SAMe, in spite of the changes, significant changes in gene expression, did not produce congenital malformations and did not produce any changes in the neurological development of the offspring up to day 30 or in their growth up to day actually 90. In one of the pathways, because we looked at the different pathways changed by SAMe, one of the pathways that were changed in both genders, but again differently in males and females, was the VEGF pathway, which is a very important pathway in the development of the brain in all animals. So if we summarize this prenatal study, SAMe treatment caused significant sex-related differences in the expression of genes in the frontal half of the brain, with differences between genders. Prenatal VPA exposure of the newborn mice did not induce any significant changes in gene expression, but pre-treatment of the SAM treated by VPA corrected or prevented actually the changes produced by SAM. And this is just the measurement of weight between the groups, controls and SAM treated. And in summary, I would not repeat the summary because we summarized both parts. But to conclude, VPA is a histone deacetylase inhibitor and can induce DNA demethylation, while SAMe is a principal methyl donor for the majority of methyltransferases that modify DNA, RNA, histones, lipids, and proteins. SAMe induced major changes in the gene expression only when given prenatally and not when given postnatally, and there is an antagonistic effect of VPA versus SAM, and the real problem is, can these agents be used as epigenetic modifiers in ASD, in human, at least the use of SAMe, and perhaps in other diseases. I must end by the fact that SAMe is used as a treatment for major depressive disorder for the last couple of years in human, but it is generally used as an adjunct therapy and not alone. Thank you very much.